Good afternoon, everyone. Okay, so I'm pleased to be able to share with you today some work that my research team has been working on for about the past decade on trying to advance the science and practice of plant translocation or plant reintroductions to improve the recovery of at-risk species. And I want to acknowledge a number of collaborators in this project, most importantly those from the Center for Plant Conservation, which is based out of the San Diego Zoo, who've been part of this effort with us for a better part of the past decade. So plant translocation, plant reintroductions, we've heard these words throughout this past couple of days. These are basically the intentional movement of plants or plant propagules for conservation purposes. And there's a broad spectrum of activities in plant translocation and plant reintroduction. And that's what I'm showing you here in this particular graph. And the space that we've been working in and what I'm going to talk about today is this space highlighted by this orange box. And so focusing on reintroductions within the historical indigenous range of a species. And that can be either establishing new populations in protected areas or augmenting existing populations, either to bolster genetic diversity in those populations or to increase population size in an effort to try to reduce that species extinction risk. So as a conservation practice, plant reintroduction has really been increasing over the past couple of decades. And if you look in the scientific literature, there have been a number of important reviews documenting plant reintroductions all over the world. So some more recent reviews have been published in Australia, documenting over 1,000 reintroduction projects over the past 4 decades. There's a nice review based on Chinese reintroductions in China. And then more recently, a group in Italy has put together a database of reintroductions, and that's well into the 1,000 now as well. So there are literally thousands of projects across the world of trying to reintroduce species back into the wild in an effort to reduce their extinction risk, but we still know very little about their long-term success rates. And more importantly, what are the sort of drivers of these reintroduction projects in terms of what's determining their success or failure? And so we've been putting together data sets in the United States based on plant reintroductions, again, for about the past decade or so. And we've teamed up with the Center for Plant Conservation, and they basically were putting together their own data sets. So we now have both of our data sets merged in an online database that's publicly accessible at saveplants.org. And this reintroduction database has about 480 projects representing 187 different plant taxa. And it's open to the public, so you can look at the registry and see which species are being reintroduced. You can look at information about those projects and specifically how those reintroduction projects were designed. So I just wanted to point out this important resource. And the data that we've been analyzing is drawn from this particular database. And the analysis I'm going to present today is really based on some projects submitted by a number of different collaborators and project partners, and I'd like to acknowledge those in this slide. And importantly, I'd like to acknowledge a couple postdocs who've been instrumental in synthesizing and analyzing this data set. Our postdoc at the Missouri Botanical Garden, Oyomawar Osuzawa Peters, and then Joe Bellis, who's a postdoc at the Center for Plant Conservation. And this is a look at the CPC database that we put together. And you can see that reintroductions, again, are not sort of spatially, they're unevenly distributed across geographic space in the United States. And I want to point out a couple of things that's interesting in this data set. So a majority of the projects occur in 2 of the world's biodiversity hotspots. Hawaii and the Micronesia, Polynesia biodiversity hotspot, about 40% of the projects I'm going to present today are Hawaii reintroductions. And then we have a number of different projects in the database from subtropical Florida. And that's in the world's newest declared biodiversity hotspot, the North Atlantic coastal plain. And so I think there are a lot of things we can learn and lessons we can learn from these projects within these tropical and subtropical regions that are applicable to this particular group that's focusing on the Caribbean. So this is a look at the growth forms in our database. Most of the reintroductions are perennial herbaceous species, but we have a fair number of woody shrubs and trees. And I want to point out that most of the trees are all tropical wet forest or dry forest trees in Hawaii, and most of the woody shrubs are from subtropical systems in Florida. And we have some really underrepresented groups. Graminoids, very few documented reintroduction projects with rare grass species. Non-vascular plants are virtually non-existent. A handful of ferns and vines. So if you're really interested in working in these sort of underrepresented groups, is a great place to really add some important knowledge to our understanding of plant reintroductions. So we extracted about 285 projects from the database that had enough information to answer our question that we were interested in. And that's like, what is the relative importance of species traits, restoration technique, and the attributes of the locations that we're translocating a species to? What is the relative importance in determining plant translocation outcomes? And some of the data I'm going to present today is coming out in a paper in conservation biology. So if you want to learn more about this data set and this analysis, I would encourage you to go check out this paper. It's open access, so it's available to anyone, anywhere. So from the CPC database, we extracted 29 different predictor variables that we thought were important in driving plant translocation or reintroduction outcomes. And we took each one of those variables and we placed it into 3 different sort of broad categories. So we've got species traits. Those are things like lifespan, mating system, pollination mode, seed size, the rarity of the species. We've got techniques. So those are variables that are really under the control of the conservation practitioner. So these are things about how many individuals you're outplanting in your translocated population. How are you sourcing your genetic material? Are you mixing populations? Are you using single source populations? Are you using seeds? Are you using plugs? Are you using bare roots? How many individuals are you outplanting? How many times are you doing that over time? And what kinds of management are you doing at the site? So those are all things documented in the database. And then finally, the third sort of set of predictor variables are sort of attributes of the locations that are the recipient sites of our translocation or reintroduction. So these are things about habitat suitability and how habitat suitability or habitat quality might change over time. What kind of ecosystem is the translocation occurring in? What kinds of threats are at those sites? And how do threats change over time? And how old is the project in terms of reintroduction age? And I should note that our data analysis is somewhat unique in that our average age of our reintroduction projects is about 8 years, but we have projects going all the way up to 30 years old. So restored populations that are now 3 decades old. A much longer sort of dive into looking at translocation success and what are those drivers of translocation. So we took all those variables and we asked, how do they influence the different metrics in which we measure translocation outcomes? We focused on 6 in our paper, but I'm going to talk about 4 in the rest of this talk. The first is population persistence. So is that restored population, is it persisting or is it going extinct? The second one was next generation recruitment. So are you getting next generation seedlings? We want larger populations to reduce the extinction risk of a species. And then we focused on reproductive population size because that's really a measure of effective population size. So how many individuals are reproducing in the population? And so what we did is we took all those variables and we applied machine learning statistical techniques to ask which one of those variables were most important in explaining each one of these outcomes that we measure in plant translocations. So when we look at whether or not a reintroduced population goes extinct or not, we found a couple of predictors were most important. One is the founder size. This is the number of individuals that's being outplanted in your reintroduced population. So that was the most important predictor in determining whether or not your restored population went extinct over time or not. And the second most important factor was the quality of the habitat. So if practitioners thought that the quality of the habitat changed from the time that they started their reintroduction project to their last monitoring date, that turns out to be a really important predictor of reintroduction success. So something you won't see our species traits don't really show up as being important in predicting the persistence of reintroduced populations. So this is looking at the relationship between founder size. On the y-axis, it's logarithmically scaled here, and the probability of extinction. And we see this inflection point at about 100 individuals. So those reintroductions that outplanted more than 100 individuals had about a 20% lower rate of extinction over time than those that planted less than 100 individuals. And we see something similar when we look at next generation recruitment. So this is, again, the probability that that restored population is recruiting new seedlings from that initial founder population. And again, we see this sort of inflection point at a little over 100 individuals, and we see 20% greater recruitment rates for those reintroductions that started off with more than 100 individuals. We also find that founder size creates more resilient reintroduced populations. And so this is looking at the interaction between whether or not the habitat quality has changed over time at those reintroduction sites and the size of the founder population. So in populations where there's been a negative change in habitat quality, the habitat has degraded over time since the start of the project for some reason. It could be invasive species, it could be a disturbance. Those populations that were founded with more than 100 individuals tended to have a much lower extinction risk than those that were founded with about less than 100 individuals. So again, larger populations are more resilient to changes in habitat quality. So this is looking at the relative importance of our predictors, our top 10 most important predictors for 3 different metrics of population size. And again, we see founder size is coming out as being one of the most important predictors, but species traits, which are in this yellow, in the yellow color, are kind of coming out as being important in a couple of these different metrics of population size. So the life form of a species, whether or not it's an herbaceous species or a woody species, turns out to be important, and I'll show you what that means in just a moment. And then seed production rate, so how well a species is producing seed. Is it a strong seed producer or is it a weak seed producer? And so what we found with reproductive population sizes is that perennial herbs tended to form populations that had lower number of reproductive individuals, or they typically didn't have any reproductive individuals at all in their population. This was particularly true for herbaceous species that were poor seed producers. And so slow-growing, temperate herbaceous perennials that have low reproductive output tend to form populations that have low number of reproductive individuals. On the other hand, something that surprised us is we found that woody species were forming some of the largest reproductive populations in our data set. And when we looked a little bit deeper, it turns out that many of those are being driven by subtropical subshrubs and shrubs that have fast growth rates and tend to be really strong seed producers. And so those species tend to form much larger reproductive population sizes. Another factor that was important in explaining reproductive population size was the number of threats. And so threats could be things like invasion at a site. It could be herbivores or predators. It could be disease. And the more threats there were at the reintroduction sites, the more likely it was those reintroduced populations had higher probability of not having any reproductive individuals in the population. They also were less likely to form medium to large populations. So threats were really important in determining this important metric of reproductive population size. So I think this analysis has some important implications for best practices in rare plant reintroduction. So in the Center for Plant Conservation Best Practice Guidelines, which is freely available, I think it's a great resource if you're designing a reintroduction program with endangered species. I'd highly encourage you to check it out. It kind of goes through all the steps of how to structure your reintroduction project. We recommend that you should have a minimum of 50 individuals, but I think I'm going to recommend, based on this analysis, that we have a minimum of 100 individuals in your founding population size as a minimum starting point. Something else that came out, and it's not surprising, but threats are important, and you need to reduce those in your reintroduction program at the time of outplanting, and you need to manage them over time. And so what happens is in the longer timescales of these projects, in the order of a decade or more, you start to see new threats emerge at these sites, and they need to be managed and contained in order to ensure the sustainability of that reintroduced population. Something else that came out is that it's really important to match the niche of the species to the reintroduction site. And we've got a whole nother set of analyses we're doing on looking at how practitioners and how we do these reintroduction programs, how do we choose sites, how do we gauge the niche of a species and whether or not it matches the site. And that's sort of a forthcoming analysis we've been working on. But something else that came out is that we really need to make sure that we're managing our sites that we're reintroducing to. That's going to be critically important. And something else positive from this is we found about 80% of the projects in this data set that we analyzed still persist on the landscape today. So a large number of reintroductions are contributing individuals that are both flowering, they're contributing to gene flow within that species. And just by having more occurrences on the landscape, you're naturally reducing the extinction risk of that species. And so I see that as a really positive outcome from this data analysis. And the other thing is that there were many different predictors that didn't come out as being important in our analysis. So how rare a species was didn't matter. And we found that ecosystem type wasn't important really either. And so what that suggests is that these reintroductions are a tool that can be applied broadly across almost any type of species in any type of ecosystem. And we do have success stories in all of those different systems. Thank you.